So next up, we have Natalie Artzi, who's an associate professor at the Brigham and Women's Hospital, Harvard Medical School, also with an appointment at the Institute for Medical Engineering and Science at MIT. Her work focuses on development of new treatment modalities for solid tumors, including GBM.
Thank you very much, Forrest, and I'm so excited to be here today. My lab really focused on GBM starting about 7 years ago, when we realized that we've been working on very important problems, including many peripheral tumors. But in fact, the tools that we developed in the lab, some of the biomaterial-based tools, are best suited to make a significant impact on tumors where barriers to delivery are so significant. So today I'll tell you a little bit about that story and what we do in the lab in the context of GBM. But one thing that I think you should pay attention to, and it will be the theme across my talk, is that we really care about the time parameter. You've heard many talks today where we try to kill aggressive tumor cells, and that's the short-term or the progression-free survival perhaps, but we do need to train the immune system to help us eliminate the tumor. Maybe that will be then the long-term overall survival that we'll be seeing. So we try to use materials to control not only the presentation of drugs, but also the duration, how long the drugs will be in the brain in order to educate the immune system and get some immune memory. So these materials can do a lot of things. One thing that they can do, and you can see here in green, is a cross-section of an adhesive hydrogel we used to localize the delivery to the brain right after tumor resection. And in red or pink, you see here microparticles that break into nanoparticles that deliver, in this case, nucleic acid. And when you stain them nicely, you get a nice bouquet. It's one of the things that we can do with those materials. So let me tell you a little bit about what we do in the lab. By designing and understanding the interactions between materials and drugs, we can develop better drugs. We developed different tools across length scales, from nano and microparticles to macro scale materials like hydrogels that I'll tell you a lot about today, and as well as microneedle patches that are really cool. They are like a Band-Aid that you can apply with hundreds of projections of microneedles that are polymeric. They can release drugs effectively at a certain location. We use them both for internal organs as well as for skin delivery. And they're non-painful. Even when you apply them on the skin, because of the small micron scale, they don't reach nerves. So that we have a lot of things that we've done with those microneedles, both for delivery and diagnosis. But really, by understanding structure, function, properties of materials, and their interaction with the immune system, we can engineer the immune system very effectively with the goal of training the immune system to get long-term memory, like a vaccine. So one of the projects that really represents some of this thinking in the lab is presented here. We essentially got an ARPA-H grant last summer, where we, in collaboration with colleagues at the Wyss Institute, we discovered a drug. This is from Don Inberg's lab, an RNA drug that activates the innate immune system. It's an RNA sensor, or the RIG-I pathway, where we use DNA origami with William Xi's lab to present the drug in a certain way to better engage with the target. Then we use different nanostructures from my lab that in fact have different tropism to different organs and cells. And that allows us to shuttle the drugs to different areas and understand their mechanism of action. We may know what the pathway is, but where the drug goes and for how long it goes really affects the outcomes. And with Don Inberg, we really look early on at human chip models where we want to see what the effect on human cells is. So we leverage some of those tools now in the treatment of glioblastoma. And as you heard today, we know that glioblastoma, following resection within 6 months, despite radiation and temozolomide, shows, again, usually recurs very close to the resected tumor. We also know that at the point where the tumor has been resected, if we use a different contrast, we'll see via MRI a hollow, basically a halo that shows that the cancer cells are still there, but they infiltrated healthy tissue and cannot be removed. We also know that the tumor can be resected and still appear at another site. So the idea is that we probably need a brain-wide treatment in order to eradicate the tumor. But when we think about delivery to the brain, in order to get the drugs everywhere in the brain, we are really challenged with the blood-brain barrier. The other thing I want to highlight is the time period or the gap between the diagnosis to tumor resection, and then from tumor resection to the initiation of treatment. The 6 weeks gap between surgery and chemoradiation really facilitates the local spread of the tumor. And we thought, what if we could intervene early on, just right after the resection of the tumor? We can apply materials before closing the patient. We can apply materials locally that will release the drugs. Now we can release any drug of interest because we are not limited to the ones that cross the blood-brain barrier. And we can also prolong their release, and I'll explain to you why. So the idea is, in addition to radiation and chemotherapy that are mainly cytotoxic drugs that are tasked with killing every cancer cell, we'll also leverage immune therapy. By training the immune cells to identify and eliminate the cancer cells, those cells can then circulate in the entire body, or in this case, the brain, in the entire area, and eradicate the tumor. So that was the premise of the work, that we probably need to combine chemotherapy to eliminate the tumor as much as we can, as this is a moving target, but also start to train the immune system. And since the training takes time, again, the time parameter, we need to combine them to get rapid killing as well as prolonged education and then elimination of the tumor. Some of the tools we developed in the lab, again in the nanoscale and then the hydrogels, are very different than other materials. Our materials are designed to be dynamic, to sense their environment and respond to it. And we believe that this enables better precision medicine. Here we design nanomedicines that are cell-responsive, that were designed to deliver the stimulator of interferon gene or the STING agonist. You may know that the SIGA-STING pathway gets activated every time we get an infection. So we thought to leverage this mechanism to now alert the immune system, to recruit cells, activate them, and train them to eliminate cancer cells. But then these particles, while can be delivered systemically to treat peripheral tumors, can be combined with the chemotherapy locally in our adhesive hydrogels to treat brain cancer. And that's what we thought to do. When we think about STING agonist delivery that you can see here on the left, and as well as other nucleic acids, we know that those drugs cannot be delivered in a free form and be injected to the bloodstream because of rapid clearance and degradation and inability of these drugs to really get into cells. So in the field of nanomedicines, we use the negatively charged of these drugs to interact with positively charged particles. And this electrostatic interaction is usually stable enough to shuttle drugs effectively into cells. However, with the STING agonist, because it's such a small molecule, there's not enough avidity to create a stable structure. So we created new structures where we chemically conjugate the drug to the particles. That renders it very stable. However, such a small molecule, any modification that you do will render it inactive. So we designed actually the particle to have a cleavable linker that is responsive to cellular cues. It can be pH, redox sensitivity, or in this case, we use the catepsin-sensitive linker such that enzymes in the immune cells, in the cytoplasm, can cleave this bond and release the CDN drug, the cyclic dinucleotide, which is a STING agonist drug. So that's how it looks. It can be any core particle. In this case, we use polybeta amino ester, which is a very biocompatible material that can condense RNA or DNA molecules very effectively. And we use the catepsin cleavable linker such that in this cell, it will release the STING, the CDN will activate the STING pathway, result in type 1 interferon release that then recruits a lot of our immune cells. And that's how the structure looks. A drug that we used, the Takeda drug, it was a collaboration with Takeda, a linker that is sensitive to the enzyme. And in this case, the core particle was based on PBAE, but it can be replaced with other cores as well, if desired. So you don't need to understand all the chemistry, but just so you understand how long it takes to synthesize such a particle and drug. Indeed, what we need to do is to modify this CDN by protection and deprotection group to include the cathepsin cleavable linker, and then another set of deprotection and addition of malamide-containing linker. We now have a group here, the malamide, that can click into a particle of interest. And this chemistry enables us to stabilize the molecule on the surface of the particle. But this needs also to be degraded. So we designed a cleavable linker to degrade on its own, and I'll show you how it's happening. But then we synthesized the polybeta amino ester particle. This is a synthesis that results in interaction of amine groups and acrylate groups with modification to include thiol and furan groups to then click the malamide modified drug to our particle of interest. Now we have a very potent nanomedicine, but it needs to release the drug. So we designed the cleavable linker to have two-step self-immolating degradation to have the scarless form of the drug, to make sure that it remains active. So all of this process, does it result in better efficacy? So to really look into that, we published a paper on melanoma, breast cancer, colon cancer, and I'll show you one result in a melanoma model that I think that really exemplify the importance of the structure of the particle in dictating efficacy. And you can see that this is the untreated mice in black, and when we deliver the free sting agonist, which is extremely potent, we push the survival just a little bit. The complex that has just electrostatic interaction improves the survival some more. But all of those mice succumb to the tumor. When we use the conjugated form that is cell-responsive with the cathepsin-sensitive linker, now it's the same drug and the same dose, we now cure 100% of those mice. So we went on and took this drug and wanted to see how we can leverage that to treat glioblastoma. We developed this adhesive hydrogel that over the years we used to deliver a range of drugs. There are not many materials that you can deliver small molecules, antibodies, nucleic acid within the same hydrogel. And the hydrogel sticks to the site of interest, which becomes very important, especially in the brain. We could deliver chemotherapy, antisense DNA, microRNAs, sting agonist, anti-PD-1. And let me show you what we've done in terms of the brain. So the idea was, following resection, we can spray coat or inject this adhesive hydrogel to the brain. It will contain all the drugs of interest. It will adhere to the tumor cavity, to the tissue following the resection. And right where all the leftover cancer cells are likely going to be, we start to release drugs to eliminate the tumor effectively. The idea is that it's easy to administer. We don't have to wait, close the patient, and initiate treatment after 4 or 6 weeks. The material can adhere to a range of tissues and control the release of the drugs. And we believe that the controlled delivery is actually critical. The material is composed of 2 polymer dextran that we oxidize to form this aldehyde group, and dendrimer that has 128 amine groups. This multivalency enables very nice interaction and binding between aldehyde and amines to form the gel. But also, those aldehydes can interact with amines on the surfaces of tissues to form the adhesion. But the key is that we still have a lot of excess functional groups that can interact with drugs. And that is what enables us to sustain the delivery of drugs over the course of weeks rather than a few hours. So the work that we've done in the context of glioblastoma was done by a very talented PhD student, Michelle Dion, who actually just graduated from the HST MEM program here at MIT, who's done a fantastic job. She leveraged these tools to release drugs of interest, but to really use that to understand the mechanism of action of these drugs and create some design rules by which we can train and educate the immune system to eliminate glioblastoma. What I'll show you here can probably be relevant to many other drugs, but we focused on a combination of drugs that we thought would be very interesting. One is doxorubicin. It's a very potent chemotherapy that when delivered systemically at a certain dose can cause cardiac arrest and toxicity. But what if we can use hundreds of these doses, because we now localize the treatment to the brain, but really leverage the fact that it's 750 times more potent than temozolomide. The other aspect is that this drug can result in immunogenic cell death of the cancer, which means we expose all the antigens of the cancer cells, which we believe is important in order to activate the immune system and synergize then following the release with the activation of dendritic cells by the STING agonist. This is the same nanoparticle that I showed you before with a cathepsin-sensitive linker that can activate dendritic cells. The first responders that will, we want to identify the tumor antigens, train T cells to then become effector cells, activate them, and eliminate the tumor. We also use anti-PD-1 and compared systemic versus hydrogel-mediated local delivery of anti-PD-1 to maintain the activation of the T cells. And let me tell you already here that the systemic delivery of anti-PD-1 was not effective, while the local delivery really enhanced the outcomes we've seen with the two drugs here, doxorubicin and STING. But you can think about other combination of drugs. The key here was to eliminate the tumor as much as we can because it is so aggressive and start to train the immune system, have rapid response as well as prolonged response. So adhesive hydrogel contained the three drugs, and this is the premise of the work. We really need this sustained release. We call it phase 1 to 3, where in the first phase, there's rapid release of doxorubicin to kill the tumor cells, expose antigens, with CDN being released to start to train the immune system and recruit immune cells to the site to make it more hot. While we do that for the next 2 to 5 days, we really want the drugs to be maintained over a week to make sure in particular that anti-PD-1 will be there to prevent evasion mechanisms from kicking in and generating immune memory. So the idea is to convert this immune-suppressed microenvironment to immunoactive. CDN high release and DOX release will begin this process. Dendritic cells will present antigens to T cells. But another thing that is critically important is that CDN can potentially also recruit and activate natural killer cells. And natural killer cells can work in a complementary way to that of the STING agonist because it's non-antigen-mediated elimination of the tumor. Especially in glioblastoma that is heterogeneous, we want to make sure that we don't rely only on antigen presentation. We know that there's also downregulation and evasion of this mechanism. So having another way that is not antigen dependent, these natural killer cells can sense stress on cancer cells, particularly following doxorubicin delivery, and eliminate those particles. In the last phase, we want to make sure we prevent immune escape. So we still want some of the CDN, but make sure also anti-PD-1 is there to maintain their action and also generate immune memory. In our mind, the generation of immune memory is the whole reason why we use immune therapy, like a vaccine. If we still have only transient response, we haven't done much. So indeed, with the hydrogel, we were able to include all these drugs and prolong the delivery of the drugs and retention in the brain that we believe is key in the outcome of what we've been seeing. I will skip the GL261 data because we got very good results in GLP-21. So now I want to focus on the CT2A model, the immune-suppressed immune desert model that we, as expected, see that there's resistance to any of the monotherapies, whether it's doxorubicin, the CDN nanoparticles, or the anti-PD-1. But as we expected, when we combine the dual therapies, we start to see very nice increase in survival. But what was really cool is that the triple therapy that really made sense to us in terms of the immunity cycle and the long-term anti-tumor response indeed pushed the survival in such an aggressive model to 80% of the mice. The mice were completely cured. And we ask one, will they survive post-re-challenge to see if we have immune memory? And the other question we ask, is it really that we need the prolonged delivery via the hydrogel, or could we just take these 3 drugs and deliver them directly to the tumor intratumorally and get the same effect? So the answer is the hydrogel is absolutely necessary. Intratumoral injection of the triple therapy resulted in only 30% of the mice surviving. But the most important point is the re-challenge. When we now go and re-challenge those mice, after 90 days, contralateral re-challenge, most of the hydrogel group mice survived, while almost all the IT-delivered drugs group succumbed to the treatment. So we see that indeed the prolonged delivery is important to educate the immune system, eliminate the tumor, but also resist re-challenge because of immune memory. Now we probably have hundreds of graphs of immune phenotyping. I'll just show you a few today to make the point that we really believe that we need almost all the immune cells in the brain to engage and work in concert in order to eliminate this aggressive tumor. We see that in response to our treatment, we have many more CD45 positive immune cells. They go up in the CDN nanoparticle treatment group, but primarily in the combination groups. When we look at CD45 high, which are mainly the microglia, macrophage and microglia, we see that indeed CDN and combinations increase those as well. What's interesting is that these macrophages and microglia were converted or polarized more so into the M1 phenotype. We just heard that more than 30% of the tumors contain these macrophages and microglia that support tumor growth. So the ability to convert them to the inflammatory phenotype is critical. We see less of the M2 phenotype by the reduction of the CD206 positive signal here. But the one thing that was really curious, if we look already, it's only 3 days after the initiation of treatment, we see that these active microglia and macrophages contain PD-L1. So this means that we have a potential immune regulatory mechanism, which makes sense. We know that it happens, but very early on, which really motivates the combination with the anti-PD-1. Otherwise, the PD-1 on T cells can interact with the PD-L1 and become ineffective. And to remind you, we really want them to not only help us eliminate the tumor, but also result in immune memory. So that really showed us the importance of the combination that we chose, but also the sustained delivery. Just to show you briefly, dendritic cells indeed were activated, as we see in the combination treatment and the CDN. We see CD86 positive dendritic cells showing their activation markers, primarily high in the combination groups. We also saw that indeed natural killer cells, we had more of them and they were active in response to the combinations, and the CDN but higher in the combination. And let me tell you, before we go, instead of showing you all of the characterization that we've done, we would expect that macrophages will play a role because they're significant in the tumor microenvironment. But what about the scarce natural killer cells and T cells? And to really investigate their role, we depleted those cells. What was really interesting to see is, despite the fact that we activate all of these different immune cells, depletion of just the CD8 T cells, for example, eliminates the efficacy of this triple treatment. It really shows us that every immune cell almost is important in this treatment efficacy. Same goes with natural killer cells. They're so scarce in the immune microenvironment, and yet when we eliminate the natural killer cells, we eliminate the treatment efficacy almost altogether. So we suggest that rather than looking only at the dendritic cell T cell axis, as we tend to look at the immunity cycle, we now believe that macrophages, especially in the brain, play a significant role, both by direct tumor killing as well as by releasing chemokines and signals that will recruit effector cells like T cells and natural killer cells. And those really help us because of the difference in time scale of their activity. Natural killer cells can rapidly eliminate cancer cells in a non-antigen mediated manner. They don't need to be trained, so this can happen fast. As we start to train the T cells to recognize and eliminate the tumor and create immune memory. So we believe that this modified immunity cycle is important to take into account and can inform some of the selection of treatments that we choose to treat tumors and glioblastoma in particular. I just want to mention some cool projects that now we collaborate on with Forrest And Yufei here, Michelle worked really hard with Yufei to start and understand how the treatment of doxorubicin and CDN together enhances potentially antigen presentation or diversify the antigens. And can we use that actually as a way to provide also a vaccine or these peptides using, for example, mRNAs to encode for those to further eliminate the tumor to result in complete elimination and memory, anti-tumor memory. We're very excited about this potential, and especially in heterogeneous tumor. And one of the things we really need to consider, as Forrest mentioned, is which peptides actually are we going to choose. It seems like doxorubicin CDN does a pretty good job, particularly probably for the tumor cells that share these peptides of interest. So the question is, should we target the more scarce one that maybe we enrich for over time? So this is something we're very excited about. With that, I would like to summarize. I showed you that biomaterials can really be designed and leveraged to respond to tissue and cellular cues to maximize therapeutic efficacy. By designing nanomedicines that are cell responsive, we could enhance the therapeutic window and understand the mechanism of action of the drug. By chemically releasing those drugs from a hydrogel locally, we could very effectively eliminate glioblastoma, but more so expand the therapeutic window of those drugs such that we can understand the mechanism of action. And because we start with a much lower dose and can sustain the release, we really get a much safer and efficacious therapy. With that, I'd like to thank you for your attention, to the funding sources, the collaborators, Michelle Dion, who led this project, and some of the other people in the lab who helped her with some of the surgeries and the flow and really getting a ton of organs from hundreds of mice to be analyzed. That was really almost a lab-wide effort. So thank you very much for your attention, and I'm happy to answer questions.
Okay, questions?
Thanks. I'm just interested. You showed the model, plasticized model of, you know, a resection cavity and filling the resection cavity with your polymer, and that's going to be your model for delivery. Is that what happened in these mice, or was this a direct injection of the mice? Is there an actual mass effect from the injection itself that needs to be taken into account? And then do you have data on how far the drugs diffuse out into the mouse brains after these injections?
Yeah, thank you for the question. So now in the mouse models, we did not resect the tumor because it's very easy to cure the mice if you also resected the tumor. So we injected intratumorally. The volume of the gel is very small. We're talking about microliters. So there was no mass effect. We could inject it easily. Ultimately, in patients, we could use it in a tumor for the non-resectable tumors, for sure. But the idea would be following resection during the surgery to apply it to the patients as well. So that was the illustration of what we ultimately imagine we'll do. But I think one thing that would be very curious is in a window of opportunity trial, following a biopsy just to analyze if this is GBM, now you can get results within a few hours. You could go in and inject directly, and then a few weeks later, when surgery happens, you can take the tumor and actually learn a lot from it. And that will be a second opportunity for us to then apply the material post-resection. And for newly diagnosed patients, I think it can be phenomenal because before we really have all the immune evasion mechanisms, this can really change the landscape of the tumor. Yeah. Yeah.
Does it matter if you're generating an immune response?
Exactly.
Does it matter because you might be, if you're injecting into a solid tumor, you're targeting a very different population of cells than if you're looking at the invasive.
Yeah. So we did see there's some diffusion outside. I don't think it's very significant, but that's the reason we're training the immune cells to then circulate everywhere in the brain to eliminate the tumor. And the fact that when we re-challenge the mice and we inject the tumor in the other hemisphere and the mice can reject that was a good indication, at least, that that process works well.
Okay, other questions?
Thank you for a very interesting talk. I might have missed this, but what happens to the hydrogel? Is it biodegradable, or is it, so does it degrade over time, and that would probably increase the diffusion, I'm assuming from the previous question.
So the hydrogel degrades over time. This imine bond that forms is hydrolytically degradable. So it will degrade, and that's one of the mechanisms by which we release the drugs over time. And indeed, the more it degrades, you have less gel to prevent the drugs from going in. By the way, when we also kill the tumor, especially here in the tumor, when we did not resect it, there's less extracellular matrix and less inhibition for the diffusion. So indeed, initially it's diffusion, it will be enhanced over time as the gel degrades, and you don't need to then, of course, eliminate the gel or another procedure to remove it. It will just really degrade over time.
Thank you very much for your presentation. Is the idea that this should also target metastatic nodules of the tumors?
Yeah, it's a good question. So since we trained the immune system, as long as we trained it broad enough and the metastatic cells won't be very different from the ones in the tumor, in the primary tumor, then it should work. In cases where there are clear metastatic lesions, one could consider injecting to primary lesion as well, a big metastatic lesion as well, to better train the immune system to eliminate those as well. Yeah, so for example, if it's antigen mediated, then all the cancer cells that were killed and antigen exposed from the primary tumor, as long as they exist in the metastatic cancer cells, will be eliminated potentially using this treatment. If they are very different, and that's part of why we want to look at those peptide vaccines in combination with this treatment to account for those that maybe are slightly different than the primary tumor.
Thank you. Really cool and interesting. I didn't really get what's still missing to try this in humans. So what are the next steps? And then would human cells be viable in this hydrogel? So would you be able to deliver cells as well, like CAR T cells, for example?
Yeah, so a few questions. So in terms of getting this to patients, there are a few STING agonist drugs that are still in clinical trials, not for glioblastoma, but for other tumors. Actually, there's one now Northwestern is doing for glioblastoma. So we're looking into those, and it's very interesting what they'll get. Again, usually they use the free STING agonist, no vehicle, so we know the results can be very different, but still interesting to see what they're getting. Doxorubicin is also used by Northwestern. It's delivered systemically, though, with opening of the blood-brain barrier via focused ultrasound, another very relevant trial for us to watch. We believe the combination of everything we're doing should be better, especially because of the sustained delivery that we show, at least in the mouse model, is very, very important. And then the second question that you had? Yeah, cells can live in the hydrogel, not necessarily this formulation. We have many other formulations. So if you think about CAR T or CAR-NK or any other idea, those potentially can be delivered using the hydrogels in combination with other therapies.
Okay, so we've got a couple of online questions. So Monica Zamish asks, very nice work, 2 questions. Do you know the antigen specificity of the immune memory cells? So you want to answer that one first?
Yeah, so in terms of antigen specificity, we're now running OVA models to look more into that. But then the work that we do with you also, Forrest, will shed a lot of light on that. So yeah, hopefully we'll have more results soon.
And her second question is, do you think this combination therapeutic approach could be useful in the context of other aggressive cold cancer types, like PDAC, for instance?
Yes, yeah, the answer is yes. We believe it will work in other tumors as well. We worked on breast cancer, not breast cancer, colon cancer, and melanoma. Breast cancer is probably the colder out of those. And even just the CDN with the anti-PD-1 worked pretty well. We believe that when we add other therapies like chemotherapies and others, it will work even better. And something that we're really interested in is to understand how this works with radiation and other clinically available modalities that can expose, again, more antigens and recruit more cells, immune cells to the site.
Rakesh Jain says, congratulations on developing this elegant approach. A major problem with the gliadel wafer was that the drug was washed out rapidly from the blood vessels around the post-resection brain cavity. Do you anticipate this problem with your gels?
Yeah, thank you, Rakesh, and that's a very good question. We've done some of this work in collaboration with Henry Brem from Johns Hopkins, and he was excited about this collaboration primarily because we showed that with the hydrogels, we can now prolong the release rather than get an immediate washout, almost like a bolus injection of the drug that happened with the gliadel wafer. So that's why this chemically conjugated drug and the fact that we have a gel that has so many functional groups that can hold these drugs place, and as it degrades, it will release the drug, is critically important in the design. Otherwise, we won't see much of an improvement like we've seen here in the IT injection of the combination.
Excellent. So last question is from Divya Sinha, and she says, thanks, Natalie. As you mentioned, the blood-brain barrier is a hurdle to GBM therapeutics compared to other tissue tumors. Can you review if and how this is addressed in the nanomedicines, hydrogel, cell-responsive structures developed in this work? And could the application from in situ to IV depend on this and affect the response?
What was the second part?
So how does the blood-brain barrier affect basically your therapy? And that'll answer.
Yeah, so to really overcome the need to cross the blood-brain barrier, we simply place the hydrogel right where the tumor was. So that eliminates that. But the presence of the BBB will also affect the ability of peripheral immune cells or other factors to potentially enhance outcomes in the brain. So that was nicely exemplified in some of the work I didn't show today, that systemic delivery of anti-PD-1 that we thought can enhance migrating T cells from the periphery to be more active, it didn't really pan out so well either. So we definitely think that some of the brain is almost like a separate box with its own immune system. The cervical draining lymph nodes are very important and probably communicate there between the results in the communication between the brain and the periphery, but still the periphery is so much more isolated. And again, if we think about combination of systemic therapy with some of the local therapy, we'll probably need to open the BBB using focused ultrasound or lead technologies and some other ways to leverage both compartments.
Awesome, thanks.
Thank you.
All right, so let's thank the speakers from this morning again.